Next, we'll hear from Lee Puckett and Ayuda Rananen. They're mentors to Dr. Ravender Abrol, and we'll be talking about the determination of ligand binding sites.
Hello everyone, welcome to our poster presentation. I'm Lee and this is Ida. And this is our doctor, Dr. Abrol, who has a background in chemistry and does research in medicine. So basically our project was we created a database of natural emitter compounds and looked at the protein and ligand interaction.
So Lee and I docked two different molecules to do two different proteins, and we visualized this on the computer programs, visual molecular dynamics, and Chimera, but we'll go into that later. And this type of visualization is particularly helpful in the early development of new drugs, but because of the sheer number of potential drug-like molecules, something like 10 to the 50 number of molecules that could be turned into drugs, we need to find a way to better organize our current list of molecules. So in this particular study, we created two databases, one based on molecules that are found in natural sources and another based on bitter tasting molecules. And we are planning to use these databases to help further researchers if they want to find a particular drug that suits their research.
And so I focus more on the natural compound side. So natural compounds provide an easy way to give us a various amount of benefits. However, the actual mechanisms of creating those benefits we're not actually sure of. They also provide a privileged molecular scaffold, which means, in other words, that our bodies are already used to taking in these natural compounds, which are found in a variety of sources, such as herbs, vegetables, seeds, spices, and all of which have a variety of effects, such as cancer prevention and clearing the GI tract.
I focus more on bitter molecules, which have been known for traditionally as a signal of an element's or substance's toxicity, but recent studies have also shown that bitter molecules produce beneficial effects in humans. Amargentin, for example, is shown to induce apoptosis in cancerous liver cells and therefore helps the treatment of liver cancer. And also, more research also shows that bitter receptors are not only found in the mouth, but further down in the gastrointestinal tract and in the lungs and in the brain, which further increases their potential to treat people.
All right, so first we created a database, one of natural molecules and one of bitter molecules. We came up with 50 molecules so far of each. From the molecules, we started researching. We looked into specific scientific journals and PDFs and research articles online. And from there, we collected a bit of data of each. So, of course, the name, the source of the molecule, the zinc ID. Zinc is a formal database that's already set online that includes information on molecular weight and other properties of the molecule. We also put a two-dimensional structure, and protein target, and the known effects. This specifically is my natural molecular database. Here, highlighted in red, you guys can't see this, but this is my ligand of choice, which is capsaicin. And so the capsaicin is usually found in chilies, as noted here, and so forth.
And then here's a small excerpt of my bitter molecules database with diphenyl here at the bottom. And we chose this molecule because it's one of the bigger bitter molecules and is therefore easy to visualize in the protein as we did. And for the actual docking, we took two molecules, capsaicin, the natural molecule, and diphenyl, the bitter molecule. And we chose two proteins to dock them to. So for diphenyl, we chose taste receptor 2, member 38, abbreviated as TAS2R38. And for capsaicin, we did TRPV1, the protein. And for actual docking confirmations or configurations, we used a web server called SwissDoc, which can be found online. And then the actual configuration of the ligand was found through zinc ID, as Lee mentioned earlier, and the configuration for the protein was found through different studies.
So we actually wanted to visualize the bindings of these from Swiss docking. So from Swiss docking, we used two programs, Chimera and Visual Molecular Dynamics. or otherwise known as VMD. And Chimera is used basically to visualize these hundreds of conformations that were given from Swiss docking. From our research, we were giving views of the electron density of where these compounds possibly bind to, but not the exact conformation. So what Swiss docking does, we're able to see in Chimera that the exact location of these compounds. And here, I don't know if you guys can see, but this is like a brief model of what our research is. So this was Chimera specific. We actually went through hundreds of these clusters, and we determined the exact, or the closest to what we predict as the exact conformation of this.
And the most probable binding site was found through its actual place on the molecule, or on the protein, and on the greatest release of Gibbs free energy.
So after we found the conformation which we thought was most accurately represented the actual conformation, we put the protein and ligand into VMD, which is a better visualizer. And so this is an example of TRPV1, which is a signal detector that detects both chemical and physical stimuli. And as you can see here, TRPV1 is usually found in a group of a cluster of 4. Capsaicin itself increases metabolism, reduces nerve pain, and prevents cancer, and also provides a burning sensation. So from here, we were able to predict the exact amino acids from which capsaicin binds, or which capsaicin interacts with TRPV1 within a 3-angstrom range. So the amino acids are listed here.
And then here is diphenyl within the protein TAS2R38. And TAS2R38 its 2 predominant alleles are the determining factors in whether or not an individual can taste PTC, which you might remember from your science class with the bitter tasting sheets of paper, or the no taste sheets of paper if you don't have the allele. But here you can see diphenyl inside of the protein, and you can see it's clearly toward the top of the protein, so it's most likely to bind pretty close to the top, and it's also well within the protein as shown in this picture.
So we hope that our project continues. Someone will pick up this database and continue adding on more and more of these molecules and continue binding and knowing the exact conformation of these ligands to the protein. Because this helps immensely with medical research, because it creates an easier way to access all of these and helps in the development of future medicines. So we'd just like to acknowledge our support of both Cedars Sinai, the facility is amazing, and also the National Center of Advanced Translated Science, which provided the grant for us. Thank you.
I just would like to understand clearly that basically you identified a new ligand binding site of this protein.
Yeah. Yes, well, we can't actually confirm that that is the exact conformation and that those specific amino acids bind with it, but it gives a much closer representation of it. And also, you're sitting kind of far, but we can actually determine like which of the amino acids are within a certain range of the ligand. So it gives a much more clear representation of the interaction between the ligand and the protein.
You talked about capsaicin having an anti-tumor activity.
Yes, that was actually part of my research in a Nature document, which is where I got the electron density. And yes, capsaicin is actually known to help prevent cancer resistance.
So we should all be eating tons of habanero.
I'm not saying that, but it has, one of the known effects is to have cancer prevention. Actually, most of these compounds have cancer prevention, even some of the bitter molecules.
When you talk about something like capsaicin having multiple different activities, do you think that that is due to interactions with just one binding protein, or do you think that those are related to interacting with different proteins?
Could Could you rephrase the question one more time?
You've identified how you think capsaicin interacts with one protein in the body. Does capsaicin interact with many different proteins? And do you think that it's different activities are due to interacting with different proteins or just with interacting with one protein?
Okay, so in our research we found that a lot of these ligands, if we know the known protein that it targets, there's actually multiple proteins which the single compound actually can connect to. And it goes back to what Dr. Abel taught us, the multiple log, multiple key model, which there's many different conformations of which capsaicin can bind to just to this particular one, to this particular protein, which also means that it can also bind to several other proteins that we don't know yet, but with further research, hopefully we'll be able to determine these exact proteins and then determine from those proteins which create the preventative cancer effects, which prevent increased metabolism, etc. Eric?
Can bitter molecules also be found in nature? I know you're saying the difference between natural molecules and bitter molecules. Are you saying that bitter molecules are synthetic, or?
Oh, no, they can be found in nature as well. They're actually, a lot of my bitter molecules in the database actually come from natural resources. So it's possible for the two to overlap.
Okay, thank you very much.